Every day, thousands of patients are prescribed identical doses from the same semaglutide pens, following a single standardized protocol. Yet the biological outcomes are anything but uniform. This chart displays data from a 2025 study in the journal Obesity, which tracked patients on a fixed 2.4 milligram weekly dose of semaglutide. Within 4 months, these outcomes split. Patient A loses weight 58% faster than patient B. The cause of this variance is a single variable, the Rs6923.761 variant on the GLP-1 receptor gene. Patient A carries the AA allele, while patient B carries the AG allele. When a patient fails to respond, clinics often assume poor compliance or lifestyle factors. In reality, the protocol itself is mismatched with the patient's genetic hardware. Without accounting for these variants, trial-and-error prescribing frequently hits a biological ceiling, leaving patients on ineffective treatments for months. This biological variance is the unseen pressure point in a $150 billion peptide market that relies heavily on cash pay experimentation. Boutique longevity clinics charge significant fees for access, yet the majority of their protocols lack any level of genetic personalization. For the industry to navigate increasing regulatory oversight, it must adopt evidence-tiered data platforms that justify prescribing through genetic guidance. The industry is moving from symptom-based guessing to predictive genetic matching to secure its medical and legal standing. Traditional pharmacogenomics is currently the primary standard for personalizing medicine. This system maps the CYP450 enzymes in the liver to predict how a patient will metabolize small molecule drugs, like antidepressants or statins. Some clinicians are now attempting to use these same legacy CYP450 tests to guide peptide therapeutics. This approach is biologically flawed. Peptides bypass the hepatic CYP450 metabolism system entirely. This schematic shows the actual clearance mechanism. While small molecules reach the liver, peptides are dismantled by proteases in the bloodstream and tissues. Because the clearance pathways are distinct, traditional pharmacogenomics panels provide no clinical prediction for how a body will handle a peptide. Effective peptide genetics requires a different framework, focusing instead on receptor shape, pathway signaling, and protease activity. Small molecule genetic testing is ineffective in this context, requiring a shift toward a peptide-specific biological paradigm. A peptide's effect is determined by how it binds to a specific cellular receptor to trigger a biological response. A single nucleotide polymorphism can change the physical shape of that receptor. If the mutation occurs at the binding site, the peptide fails to connect, and the intended signal never initiates. Mapping a patient's receptor and pathway genetics provides the necessary biological context to predict the response to a specific therapy. This creates a data problem. A human clinician cannot manually cross-reference millions of genetic variants against biological pathways for every patient. Automated knowledge graphs, built upon vast biomedical databases, provide the computational architecture needed to solve this exact problem at scale. The first step involves ingesting raw DNA sequence data and isolating specific patient variants. This diagram shows the algorithm mapping the variant to the specific cellular receptor or enzyme it encodes. The system then links that receptor into the broader biological pathway it controls. Finally, it identifies the therapeutic peptide that modulates this exact pathway. These color-coded tiers rank the strength of the connection, from direct clinical trials in Tier 1 to pathway inference in Tier 3. Knowledge graphs translate millions of genomic data points into actionable, evidence-based clinical decision support. This technology is entering a regulatory environment that is increasingly focused on longevity clinics and compounding pharmacies. The FDA is targeting off-label interventions and protocols that have operated without sufficient documentation. To remain compliant, clinicians must move away from generic protocols towards prescribing that is justified by genetic data and cited research. Clinicians now face a regulatory landscape where algorithmic precision is a requirement for operational survival. A current limitation of peptide pharmacogenomics is that many compounds still lack large-scale prospective data. Structured clinical cohort programs are the mechanism being used to capture this missing longitudinal data. This cycle starts when a clinician prescribes a peptide based on the algorithmic genotype match. Next, the system captures the patient's real-world biological response over time. Finally, this clinical outcome data feeds back into the knowledge graph to refine future confidence scores. This continuous feedback loop replaces anecdotal observations with an empirical evidence base. The era of standardized peptide protocols is ending, along with the model of cache-pay experimentation. Genetic variant mapping, scalable software platforms, and rigorous longitudinal tracking are now converging into a unified, evidence-based architecture for precision peptide therapy. The integration of genomics and data platforms transitions peptide therapy from a field of guesswork to a documented clinical science.
Thanks for watching our show.
If you like the show, please subscribe, comment, like, or share with a friend or colleague to keep the mix moving.
Thanks again, and have a great day.